As people are coming on, I'll just talk about where AGP and the Center for Genomic Pathology stands right now. We're finally moving the whole slide image server, which used to live at the mouse biology program in Davis. It was kind of a pain for us because we'd have to go to Davis to get our slides scanned. We're moving it to Sacramento. We're upgrading it to the newest version, which has pluses and minuses because, as you may know, we had some custom programming on top of ours that we're going to lose, and we're going to have to see if we can get some of that back. I think some of that functionality is in the new version, so I'm happy to go to the new version of eSlide Manager or whatever they call it now.
Yeah.
So you all should still have logins. If people don't have logins to my my eSlide Manager server, there is a lot of content on there, which is CGP, AGP. Some of these are slides you all have shared with me in the past. Others are collections from the Mouse Models of Human Cancers Consortium and the Consensus Conferences. And I'd like you all to have access to that again, which you should still right now, but there will be a short downtime when we switch over. We're also moving the educational content, the mouse biology, mouse pathology courses that we assembled, and we're trying to revamp those a little. And that's where all of you come in and helping me revamp those. May assign you to do an audit of a specific section that's your area of interest, and you can tell me what's completely hopelessly out of date and what's still really good, and then we can fix it. So everybody, as you're logging in, be warned, you're going to be hit up for this service, which is part of being part of the educational mission of the Center for Genomic Pathology that we all, I think, believe in. These conferences, and Jerry, you can tell me if there's any part of the meeting today that you don't want me to record, because we have started recording these and posting those on the website. Some of you have seen that. We started that with the session that we organized on mouse models for COVID. And we're going to try and continue that. That was always sort of the plan, but I was technologically limited a few years ago. And so we're finally getting that up to speed, in addition to having the website interface be more seamless. You can see Tyler Kim Hayes on here is online. He's my programmer extraordinaire, and for technical questions, I rely on him. So he's the one actually doing a lot of this work, and it's great to have him on the team. So quick introduction to Tyler. And I think it looks like we're just a few minutes past start time, but looks like we've got a quorum of participants. And so I'll do my usual preamble, which I alluded to, which is, Jerry, this is potentially proprietary information, and everybody online understands that and is not to share what they learn here without your permission. You can give carte blanche permission now, or you can give permission by email later, or you can say, please do not share this because this is unpublished data that my co-investigators would not want out there. But that is the assumption, is that none of this is to be shared without permission. And with that, I turn it over to Jerry, who promised us that he would first prove to us that we all have job security.
Yes. Hopefully, I don't think it should be, this one should be too difficult. First of all, thanks, Sandy, for having these forums. I very much enjoyed them. I tend not to be too chatty in them, so, and I think I last presented a year ago. So as a brief introduction, I'm Jerry. I think as I always say, I'm Jerry number 3 or Jerry number 4 in this crowd. The number of head turns I always do when people are talking and realize no one's talking to me is kind of funny. But I'm currently at Jansen. I've been there for 5 years. I support a lot of different programs, immuno-oncology programs in particular. Before that, I did my training in Boston. I worked with Rod Bronson and did a lot of mouse model work at Dana-Farber Cancer Institute with Ron Pino. Anyways, so I am, first of all, yes. So even though much of what I do is immuno-oncology studies, so, at some future forum, I'm happy to talk more about the in vivo studies for CAR Ts or bispecific T-cell redirection studies or syngenetic models. Today I wanted to kind of share some very introductory work that I'm doing, not specifically in the IO space, but it's early enough but of broad enough interest, and that's additional characterization in the APC min mouse. So you're free to share everything I talk about for this. I'll show antibody reagents. Feel free to request the antibody list. Basically, I'm doing this for half selfish reasons. You know, I want to show the rationale for what I'm doing, but also because the pathways that I'm looking at are so tried and true. I would like to hear, solicit some input if people have some insights as to some staining patterns or recommendations for antibodies. So a bidirectional exchange is very much welcomed. I'll try to just show maybe 20 minutes of didactic slides, but then do a a free-form image scope surfing to talk about findings and discuss just to discuss observations. There's one in particular that I could just use a couple of pairs of eyes on as well. So that will be one of my objectives when showing the slides.
All right,
okay.
Great.
So yeah, job security. So let me see if the slide sharing works. And in so there's a there's an image generation. There's more and more AI technology in which they try to help you, for the visually impaired in particular, identify what images you're importing into PowerPoint. So it's called alt text, but it's an automatic AI-based image generation. And it's so frequently PowerPoint has been playing pathologist alongside me and telling me what I'm seeing when I import the images. So it does a real remarkable good job at some of the standard images. For instance, I just grabbed this from the internet, and again, it's not predetermined. It basically is generating this caption de novo. So rodent looking at the camera, which is pretty impressive. That it's just not just a mouse. It's actually a rodent looking at the camera. So it does pretty well in recognizing standard images, microscope on a table, even more complex images, a person standing in front of a microscope. But then you can start to see the limitations, but PowerPoint continues to want to play along. So it gets pretty amusing at times. So this one you can kind of understand, a picture containing indoor microwave sitting oven. You kind of can see how it got the microwave oven part of things. And then there are really admirable attempts to identify pathology images like this one, a close-up of a coral. I think that's fantastic, actually, for it to recognize, kind of a coral-like pattern. This is, of course, an adenoma. That's the subject of my talk. But after that, it's been falling apart. I get a lot of fabric. I guess, herringbone patterns and things can kind of look like fabric pictures, but then it eventually falls apart completely. But it continues to tell me if this is a picture containing food, water, view, sitting. This is a close-up of an animal. This is a picture containing map and refrigerator. This is a picture containing food. And this is a screenshot of a cell phone. So suffice it to say, I don't think we'll be surrendering our jobs over to Microsoft PowerPoint and its image generation skills just yet, at least this year, maybe next.
A couple of years down the line.
Yeah. If it starts recognizing tissue, I would be very impressed. Just like, if it could say, oh, this is normal colon or something, that would be pretty remarkable. I wanted to tell a narrative, and in particular, this boils down to characterization of further characterization of APC min mice. As I understand that the APC min mouse turns 30 this year. The initial descriptions by Bill Dove were in 1990. And I think I first encountered them in taking the JAX CRC course in like 2004, maybe, or something. But anyways, I just wanted to start with why, I've worked with the APC min mouse in the past, but recently had the pleasure of being able to pick it up again in order to study it further for some downstream programs. Again, this isn't my my main focus, and again, this isn't going to be a science talk per se. It's just tool development and observations, and just to show you what direction I need to take this project into. So I'll talk about the efforts in-house to work on colorectal cancer interception, then just talk about some of the IHC staining work that I've done, and then look at some of the staining patterns and see if I can get some input. Okay, so starting with colorectal cancer interception, or actually, if you even Google the words just disease interception, Jansen is one of the companies whose names kind of like floats to the top. It's not very much in the model of hypertension and cardiovascular disease and high cholesterol. The effort is to not only cure disease, but try to identify people at high risk and find approaches to prevent it. So there's an incubator or an accelerator called the World Without Disease, and yeah, just to give you a little bit of context on that. Even if you go to the home page of Johnson & Johnson, we used to be on the very first page, but we got knocked out by COVID vaccine development. So we used to be on just as like 2 months ago or 3 months ago on the front page, but you can see the effort in disease interception, where so I'll just show you the focus of this project that I'm working on, as well as a number of related projects. But again, this is an article on colorectal cancer interception, new ways to treat it and catch the disease before it progresses. And so, various things that we're working on, which I won't talk today, is, trying to identify risk factors or mechanisms of pre-malignant progression of microbiome and cytokines as some of the mechanisms to better understand to develop interception therapies. Okay, so we can start with the let's start with the Vogelgram. So again, just to put additional granularity to that, for colorectal cancer interception, of course, we've got the well-characterized Vogelgram with stepwise, first characterized simply as a mutation, oncogene-mediated progression in which you have stepwise progression mutation events going from normal tissues. You have early adenomas, subsequent development of mutations, later adenoma, high-grade dysplasia, and carcinoma, well-described sequence. So traditionally, the approaches have been on disease therapy. If you have colon cancer, obviously you take it out. You might have neoadjuvant or adjuvant chemotherapy. If there's residual disease, if there are focal lesions, you have radiation therapy. And of course, a lot of the approaches these days are in immunotherapy as well. Now, the other approaches are preventing the disease before it starts, and you can partition that into two categories. You have disease prevention per se. Basically, prior to any detectable development of disease, you kind of try to interfere from even the very first steps happening. Maybe a vaccine that you give to everybody. It's not a very practical approach. You don't want, I don't think we're anywhere near the stage where we could give the entire population something that prevents colon cancer. So the focus has been on disease interception, in which you identify patients who have had the initial events. You try to find patients who are at high risk of developing disease later on. This is not a unique paradigm, seen in breast cancer and prostate cancer as well. And then ideally identify pharmacologic, in this case, compounds that might prevent you from developing colon cancer, even though you are at high risk for these stepwise progressions. So in terms of what we're looking at, all bases are covered. Again, so the paradigm is prevent, intercept, and cure. Okay, so yes, so suffice it to just to say there's a lot of ways to approach this effort. It can be an oncogene focused approach on particular genes or mutations, and trying to counter the effects of those oncogenes. We're looking at more global modifiers, immunity and inflammation, of innate immunity, adaptive immunity. So one particular area that I've been focused on as well is this area of innate immunity and inflammation. So I personally have an interest in the myeloid component, in cytokines and their role in DNA damage, and if we can prevent progression that way. And another robust area that we're also working on is investigating the role of the microbiome in colon cancer progressions. Yeah. So again, the really early days, but we're definitely exploring. We have some collaborations going on exploring the role of microbiome and pathogenic species in contributing. And in fact, if you look, I'm sorry, I didn't have a chance to put the credit for this figure, but if you look at this figure, these yellow and blue dots are cytokines. This is from a 2017 Nature paper. And if you look at these little brown things with the flagella, it's bacteria. So this figure incorporates the oncogenes, the cytokines, and the microbiome as driving the vogogram.
So, while you've got the vogogram up, I'll just say that, I think the vogogram is a good concept, but I think we're learning that it has one cheap problem, which is people sort of believe that it's this continuous time scale from left to right. And, we really don't think that it's very continuous. And we think there's a lot of dead ends where you might get to early adenoma and stop there and never progress. Whereas others would progress very rapidly through the stages. And it's something we're studying in breast cancer for sure right now, preparing a paper to go in. But I think it's also true in colon cancer. And I think it's I think actually Vogelstein would agree.
Mm-hmm.
The misconception here is that every time you get an early adenoma, it will march along towards carcinoma inevitably. And that's, he would agree that the time scale could be very long to get to carcinoma, or could be very short, and it depends a lot on the specifics of that initiation focus. But nonetheless, I think this is still a good framework for us all to work
Yeah,
in.
I totally agree. I think, yeah, like 90, they say, not, it's hard to do natural history studies, but 95% of adenomas don't progress, or just, they're not going to kill you. And we would love to be able to find out the signature for those 95% of polyps. So we can say, yeah, you have a dangly polyp, but it's, don't worry about it, versus that 5% in which we can find a signature that says not only, yes, we removed this, and this was one that if we hadn't removed it would have progressed. And so finding what makes those special is correct. And yeah, speaking of the other thing, the two paradigms that we're looking at, I think I'll show it. Here is obviously there's multiple different ways to get to a colon cancer. It shouldn't be thought of as a single disease, but 50 different diseases and many different subtypes. So the two, among the various flavors, the two paradigms that we study the hereditary forms in order to inform the mechanisms that develop that might be useful for sporadic carcinoma. So the two hereditary forms that we're focusing on are FAP, familial adenomatous polyposis, in which, and I'll show in the next slide, is genetically inherited disease where patients have a mutation of the APC gene. And since that is a paradigm for many sporadic carcinomas, we use that as a model. But to get to your point, the other, we look at microsatellite instability, mismatch repair deficiencies, and the hereditary flavor of that is hereditary non-polyposis colon cancer, known as Lynch syndrome, and these in particular don't seem to progress quite, stepwise. You can have a little lesion and then all of a sudden, develop into a full-blown cancer. So totally agreed with that. So
Jerry, just to follow up on what Sandy's Mark, this area too. I think this is a pathology group, and one of the things that would, I push back a little bit on the Vogelgram as well when I present sort of genetics and pathology or morphological correlates, partially because, we don't really use small and large adenomas in GI diagnosis. So, high-grade dysplasia, carcinoma in situ, or, early invasive carcinoma in a polyp or something like that, which really are things that you can correlate morphologically. So, just as a starting point, small and large adenoma, if there was a size cutoff, isn't really meaningful, because you can have a 5-centimeter adenoma that still just has APC loss. If one's correlating from the pathology side with genetics, if the morphology makes no sense because we don't really use that, or there aren't diagnostic terms to do it, you can't really correlate with something that, just isn't really something that people would agree on, I suppose. I mean, it was a nice construct, I think, like Sandy said. It's very elegant from that, but also for the mismatch repair deficient colon cancers in humans, you get flat dysplasias, all sort of, and you were alluding to those things, I think, as well. So
yeah, there's plenty of things. Yeah, and let's see. Yes, the current definitions of advanced adenomas are ridiculous. If you have 3 or more, these are advanced adenomas. If they're 1 centimeter or greater, if they're tubulovillous, they barely make a dent in predicting, higher risk. So yeah, so in parallel, we've got detection efforts. For instance, just as an aside, we engage with Path AI on ways to see, there are ongoing efforts, blood serum, microbiome, epigenetics, whatnot, in order, and histology, to find signatures that truly help identify what the advanced, what factors correlate with propensity to develop cancer. And of course, we've got issues such that the act of removing the polyp, prevents natural history. So we have to do, 5-year follow-ups and find.
That's what we have the mice for, Jerry.
Exactly. So these are ongoing parallel things. Anyways, so that's the overview. And so one of the efforts is we're going to be using mice for a long time. We're going to be using APC mutant mice as the anchor. Very limited in terms of progression. And the goal is to layer on additional things and model it more accurately, and then evaluate our pharmacologic compounds in that context. So just to combine the clinical approach with the mouse approach. So in the clinical realm, to model the APC sporadics, FAP is rare, less than 1% of colorectal cancers, but there are still enough of these patients to have clinical trials. As most of you all know, these are patients who develop hundreds, if not thousands, of polyps. They have the germline mutation for the APC gene. Nearly all of them will get cancer in their 20s and 30s, and as a result, in their late teens or early 20s, they will get a colectomy to prevent colon cancer development. As it turns out, in order to preserve bowel function, these patients will get colectomies, but it won't be complete. They'll still have a stretch of rectum, and these patients will need to undergo routine surveillance. And these are polyps that can be studied for progression. And if you go to clinicaltrials.gov, you can find 80-odd clinical studies. But it does serve as a good model for chemoprevention or disease interception, trying to find mechanisms that slow down polyp growth. Ideally, we have long-term information that prevents colon cancer formation. Difficult studies for sure, these interception type trials. And the entire axis still is being investigated. I've just listed some of the more promising avenues. So inflammation, COX-2 and related compounds showing efficacy, NSAIDs, the EGFR pathway, which is the one that I will show a little bit more characterization for. Personally involved, I won't talk about today. There is an immunology drug from Jansen. It's an anti-IL-23 called gazeltamab or Tremfya, works amazingly well in psoriasis. We were able to demonstrate IL-23 is downstream of or activated in the Th17 pathway, which in both mouse models and in human colon cancer or adenomas is present, with suggestion that it may be responsible for cancer disease progression. So we currently have a clinical trial ongoing to study whether or not this can slow down progression in FAP patients. Other studies VEGF, and there's a lot of things that don't work. Herbal supplements that have gone to clinical trials, erythromycin, a lot of things that are just kind of one-offs. But again, it's just introducing why we're going back to the APC min mouse. So anyways, what this means is, I used to use, you know, not the APC min mouse per se, but the transgenes, and we would layer on KRAS and P53 mutations. And I really never thought about studying the biology, you know, as a starting point for disease progression. So we've gone back and, you know, taken a closer look at just APC itself in order to then layer on the additional things that we're looking at, you know, whether or not it is additional genes, whether or not it is combination models such as, you know, DSS or microbiome studies. So most of you are familiar with the mouse model, but there's a recent paper by one of our colleagues, Kay, who does nicely summarize what some of the pathology for the APC min-mouse. So again, despite this having been around for such a long time, it certainly has value for us, both as an experimental model for therapy, but also for pathology analysis of drug response as well. So okay, so that was just a brief rationale. So the next part, again, nothing. This is extremely low tech by today's standards. I wanted to take a closer look at, in particular, the EGFR signaling pathway as it intersected with Wnt APC pathways. And of course, if you look at all of these models of Wnt signaling and EGFR signaling, they're single cell. It pretends that the entire world exists in a single cell. Doesn't talk about, you know, the different epithelial compartments or maturation. It doesn't talk about cell-cell interactions and whatnot. So I really wanted to get a sense of what was being expressed where. So went with, you know, some very standard IHC reagents. So I thought here in very simplified EGFR signaling pathway, both total EGFR, phosphorylated forms. There are two residues in particular that are phosphorylated, and there are rabbit monoclonals made against them. Phospho-AKT, again, it's been around for, you know, over a decade. Phospho-AKT, phospho-S6, the ribosomal protein itself. And here, among other markers, you know, just some indicators of Wnt signaling, E-cadherin, beta-catenin, transcriptionally, cyclin D1. So again, you know, just instead of just looking at them in isolation, I wanted to, the approach is one, to develop reagents, and I hate to say it, but half of this talk is going to be about just reagents, and then secondly, is starting to make some observations about it in order for us to be able to then layer on additional steps in tumorigenesis, as well as then to start testing therapeutics. So I initially thought that these would all be straightforward enough that would be plug and play, and several of them were. I was, you know, had no problems with the, you know, standard assays. And here's where anyone's input of, oh yeah, we use this or you shouldn't use that, would be helpful. And I'm also happy to contribute to that discussion, because I don't believe that people need to reinvent the wheel when it comes to, you know, I'd like to move on from having to decide which is the best antibody. But it does turn out that I'm still trying to select the best antibodies for some of these, and I'll show why. And so, I mean, just Sandy, just to pull the crowd, you know, do you in particular in your experience, have a favorite one of each of these two?
I don't know off the top of my head. Are your red ones your favorites?
Mm-hmm.
Yeah. I think that with phospho-AKT in particular, that works really well on stuff like the P10 knockout prostate model.
Mm-hmm.
In my experience, works less well when I expect to see AKT activation in the mammary gland. It's like super clear in the prostate, but a little muddier in the mammary gland. But I have a little bit less experience with some of the other. Well, we use EGFR quite a bit, and that works well. But I don't know what the clone is. But I'll be happy to kind of go back. That's another thing. That's another thing that I want the CGP website to contain, which are examples and protocols for the antibodies that we've worked up. And we've got a pretty good handle on that, but it's not as easy to navigate as I would like. So stay tuned on that, and then I'll ask you all to share experience with that. That's a huge benefit to have other people's experience getting antibodies working.
Yeah, that would be great. I'm happy to share. Share
if
this
you can
list.
share a slide, a protocol. Even we like to show a titration. Sometimes certain applications we want to change the titration slightly, and
that's not a problem, because I definitely don't want to spend excess time discovering. And to my surprise, some of these standard reagents produce drastically different results, and it's like, okay, you want to use an antibody for the next 5 years. How can you trust it? So it's just going back to control school of cell line phosphorylation controls or knockout mouse. If there's consensus between antibodies, what is in the literature. We went into staining mouse embryos and comparing, because that's where a lot of the EGFR data was, and even GFP reporter gene histologies, and I'll show you an example of which. And the other thing for phospho IHC, if anyone, let me throw out there as a side note, does anyone put phosphatase, do you know of the protocol in which you put phosphatase inhibitors in your 10% neutral buffered formalin in order to preserve phosphoproteins? There's an old protocol out there. We're still trying it, so I don't have any plus or minus experiments. But basically the same sorts of
you use it in Westerns for sure, right?
Exactly. Exactly. I just can't find good enough documentation of it, as I find vague references that you can throw in the very same things and it will improve your signal.
Well, I suggest, Jerry, you try it in a controlled experiment side by side, and then tell us if it works.
Exactly. We're actually in the middle of doing that, and hopefully I can show that to you. But yeah, I mean, you can go to HistoSearch, as far back as 10 years ago, where people say they've tried it, but I can't find a.
Seems like a good idea, but in formalin, who knows if it, all bets are off, right?
Exactly. Yeah. And also some of the IHC I'm going to show you is done on Swiss rolls, and as you know, it takes a lot of skill and manipulation to do Swiss rolls, and so especially for phosphoproteins that that might booger up the signal right then and there, and you might need to be a little bit more vigilant when it comes to preparing your samples. So even that, like for instance, D9E, I'm used to using in the prostate, but was not very happy, still not too happy with the signal that it's giving in these polyps, and maybe it's because of sample preparation. So again, I'm just throwing these out there in case people have suggestions or insights, anything to do to improve the signals maximally before we start to go to town with these reagents. Yeah, so here, here's an example of some of the struggles I had to deal with. 2 standard rabbit monoclonal antibodies against total EGFR, the one on the left from Abchem, the one on the right, cell signaling, and they were giving partially concordant results. It's this staining pattern that I want to go into in a second, but I'll just put a pin on it. Suffice it to say that if you looked at the polyp per se. These two antibodies gave similar staining patterns, but then, you know, this isn't a simple issue of dilution or concentration. If you look at the villi, this antibody implies that there's an excessive amount of EGFR signaling in the entire, you know, cryptillus axis beyond, well, I guess, the surface, as well as in the lamina propria. And this antibody doesn't. So for a while there, you know, I, you know, had the conundrum of, okay, how can I, what additional data do I have? You know, there are patterns in the literature, but that just means that people have, you know, all decided to work on one, but that doesn't necessarily mean it's right. So finally broke the tie here, where there was some nice EGFR GFP expression analysis that truly showed that, you know, in the normal axis, the EGFR signaling should be more located in the crypts, and that, so I believe that this one on the left is inaccurate. But again, you know, these are all multi-year efforts that, so it's taking taking a while, but I, you know, just don't want to read out incorrect results. So anyways, that's just a note on tool development. Again, just want to, you know, encourage if anyone has insights about particular reagents, and then just being able to get to the biology. And so I'll switch to ImageScope next. Quick time check, 12:15. Okay, so yeah, I didn't realize how long this part has taken. Sorry. And anyways, so
those are very pretty.
Yeah, we had a really good. Yeah, so this is cyclin D1, and we had an amazing, not here anymore, Swiss roller, and yeah, so basically we're you know, nothing earth-shattering science-wise, but bread and butter characterization of signaling component expression in progression of 7, yeah, you know, between 7 and 20 weeks, and for the different markers. So basically, we're still just working on the basic pattern expression. So I really wanted to get to this particular observation. What I didn't realize up front in looking at these polyps was the fact that this entire so I'm just checking to see the refresh rate. This entire lesion is not adenomatous epithelium. You know, certainly this is in the center, but I hadn't really paid much attention to this you know, hyperplastic appearing epithelial covering, and whether or not that was neoplastic as well.
Yeah, I've seen that for sure, and I've always kind of assumed that that's the more normal, although reactive, kind of stretched over the top.
Exactly. So it grew from underneath, and it's just a layering. And I think.
And when you look at the very early adenomas, they do kind of arise somewhere down underneath in the crypts. They don't arise at the top surface. Right,
and then they burst through, and I think that let me do it this way then. Right. So it wasn't until, you know, looking at, in particular, this is a nice demonstration, you know, so, you know, and I see it's been described elsewhere, but here you have cyclin D1, so you can clearly see that the you know, what is morphologically adenomatous is cyclin D1 positive, and yeah, the overlying epithelium is cyclin D1 negative. The other very nice demonstration of that is for Beta-Catenin. adjust the delay here. Again, a fairly nice demonstration that the adenomatous portion, has very clear. Sorry, just going back to normal. The beta-catenin is tethered near the membrane, so you see membranous type staining pattern. But in the adenoma per se, you have nuclear expression, and then this overlying epithelium shows still the membranous population. So that's a couple of lines of evidence that it's not.
So I had seen data on beta-catenin being somewhat labile. If it sits in formalin too long, it can actually kind of lose its nuclear signal. I don't know if you've experienced that. Okay. I think the new antibodies are better than the ones that were being used back then. Owen Sanson showed me that. He had generated some pretty interesting models. And
okay. And
that's my tidbit of knowledge. I like your nuclear staining, and it's nice to see,
it's nice basic biology that correlates with, the figures of nuclear translocation.
Right. It's supposed to, when active, be nuclear, but.
Yeah.
It isn't really, though, in human adenomas entirely, like you'd think with APC loss that it'd be nuclear, but I think it's usually, as it goes towards higher grade dysplasias and things like that, it's much more uniformly nuclear. So like here you can see it looks like there is some partial along the way, but it's not what one would have imagined, just thinking about it before you see the slides. And I don't think it's known what regulates that next step when it becomes more nuclear either. I think
because it's not, I show, there's some areas of adenoma that are nice and nuclear, but then like here are, like here are areas of adenoma. It's a mixture, and there's clearly things that regulate it more. The more by H&E more dysplastic it looks, the more likelihood that you'll see this nuclear accumulation. Okay, so time is short, but I didn't believe this result until recently. And I've already shown portions of it. But yeah, I mean, what I'm showing you is not chronologically correct. I first for phospho EGFR. This is the first observation that I made. And again, I was still at the level of, can I trust my antibodies? Is there a fixation artifact? Is this biologically real? And I think, well, one, I've now seen this with a couple of different antibodies. And two, I also have total, more confidence in total EGFR. But I now have this observation whereby activation of the EGFR pathway is not in neoplastic epithelium, but in the hyperplastic, non-neoplastic epithelium overlying the adenoma, and not in normal villi as well. So let me just show you the total EGFR one to clearly show this is the case. So so initially, if I presented this a couple of weeks ago, I would have said, do you believe this? And now this is only 40x. Well, I guess it's the same question. Does anyone know about this? Is this a described phenomenon in which EGFR signaling is enhanced not? Again, we're looking for interactions between Wnt signaling and EGFR signaling. So I expected a priori, and of course people who grind and bind have no resolution at all. But I didn't expect an increase of EGFR signaling in hyperplastic but non-neoplastic covering epithelium, not in the adenomatous epithelium and not in normal villi.
I guess I would expect that some adenomas, but not all adenomas, would have increased EGFR. And and I think this is very interesting because you can see a little bit of EGFR in your crypts, right, which probably goes along with proliferation a bit. But you're getting much more in the reactive stuff that's stretched over the top, and it does beg the question of what are those cells that are able to persist over the top. If I'm actually seeing a hint in the tips of your villi that are over to the right.
Yeah.
And I would surmise, usually those cells are kind of terminally differentiated on their way out. So I don't know. It's a funny pattern for sure. I haven't seen it, but I'm intrigued.
Was that total EGFR or is that a phosphoepitope?
This slide I'm showing you right now is total, and let me just show. And this slide right here is phospho. So it wasn't until I got both that I started believing it. And because these were historic Swiss rolls, we're now repeating the experiment with better preservation. And again, we're still tweaking the protocols to get amplified signal. But yeah.
It looks like the activator is more basolateral rather than apical, in terms of the polarized, which might actually make sense if there's something that Wnt activation induces, signaling to EGFR from the adenoma out, like on the bottom side. Hard to know, but it does look like overall protein expression is clearly enhanced. Like that looked like pretty slam dunk. And I certainly would think that would be possible. You could also, if you did single cell RNA-seq, you could also see if there's RNA for the non-neoplastic stuff, which you could follow as well.
Yeah. Again, this is just it's just a surprise, obviously the extensive history of EGFR and colon cancer and whatnot. You're just expecting it in the adenoma. So are we wrong? Are we mistaken? Is it relevant? Is it a relevant mechanism, or is it just an interesting side effect? Does it contribute to the pathogenesis? These are all issues that it seems to now be raising. Should I be paying attention to this? Do I care about this, or is it just true but not very important in tumor progression?
Jerry?
Yeah.
This is Hybrid. Just an observation here. I'm looking at this epithelium, as Marcus said. It looks really a well-polarized epithelium, and if I look at it, it looks like a well-polarized epithelium, and the expression of the this protein, whether it is total EGFR or phospho-EGFR, it looks like at the base where it is interacting with the basement membrane, the basilar lamina, and all that, where the signal is coming anyway. So do you see this pattern too? For example, if you do not have a polarized epithelium where there is more of disorganization, where do you see the staining or the labeling?
Yeah, I think that's a great question. I've actually not not until this meeting really thought about looking at apical versus basal localization of EGFR. That's not that is not at all an issue that I had really thought about. So I think that's a great next step to delve into in the literature. Let me just see if I can get some more examples of that to see if that's the case. Yeah, again, it's clear that the hyperplastic type of gland is positive, but you continue to see this basolateral expression. So yeah, I think that's a good avenue for me to look at to see if that could be a physiologically important cell sort of engagement. So That's great. That's great.
Interesting.
Yeah. So that's it. I see we're at
We're kind of at our time. Those are. I mean, I think we've all seen variations of APC and various therapeutic trials and crosses to other genes and things. And so, you know, it's really nice to have your experience with the immunostains. And I'll try and, you know, I'll try and get what we've got posted up, but then looking down the line, it'd be good to get the whole crew to share whole slide images and protocols that we can post up. And I'm happy to try and coordinate that. So,
yeah, that's great. And I'll follow up with that phosphatase experiment that we're currently running to see, because if it, again, yeah, I mean, it would be nice to be able to deal with, you know, slightly stronger signals than what we're currently.
Yep.
Doing. So I'll share that as well.
And I hope you find a good new Swiss roller.
Yeah, seriously. Seriously, absolutely. Have you tried it yourself? Oh my God,
I have done it myself. I'm fair.
Okay. Mine looks like trying to roll up a piece of spaghetti, you know, and it just turns into a three-dimensional glob. It's terrible, terrible. But anyways.
All right, thanks, Jerry.
Yeah, sure.
Any last words from anyone? We're going to see you next month. I don't have a theme for next month. If anybody would like to volunteer, please let me know as soon as possible so we can get it organized. I will be here in town. We can continue our fourth Thursday, as I've sort of stumbled into multiple months now. And if there's a lot of interest, we can go to every 2 weeks at some point. But let me know. And again, I think, Jerry, you're comfortable sharing everything you've shown here today. You didn't talk about anybody's proprietary experiments, I don't think.
Yep.
And I thank you for your presentation. I think there's some elements of our courses that could benefit from some of this as an update. So I'll be forwarding you the connection to the course section on colon of the mouse, and we can hopefully get that nicely updated.
Yeah,
So
yeah, sure.
Stay tuned on that. And otherwise, thanks everybody and have a good day. Stay healthy. Wear your mask.